naive experiment in mice in which my postdoc gave one of 3 commercially available artificial sweeteners to mice for 11 weeks. We measured everything that we could, and we were very surprised to see that each of these 3 artificial sweeteners induced an increase in the blood sugar level as compared to water or to glucose, but some people misunderstood our original publication. This glucose is not high glucose diet, which we claim is healthy. This is the small amount of glucose that is present as a bulking agent in the saccades of every artificial sweetener that you would consume. So we did this so not to compare apples to pears, okay? So this large-scale experiment suggested that artificial sweeteners induce a rise in blood sugar levels, which was very surprising, but even more surprising was that when we added broad-spectrum antibiotics to this experiment, either during or after the consumption of artificial sweeteners, we could completely negate all of these differences, suggesting that maybe the gut microbiome was involved in this process. And now me being a very known skeptic and a severe paranoid, I didn't believe this data. So I made my postdoc repeat the experiment. So he repeated it in another large-scale experiment with all 3 artificial sweeteners and completely reproduced the data. I still didn't believe it, so he repeated it a 3rd time and a 4th time. And altogether in this single experiment, we utilized over 500 mice, all giving us the same exact surprising observation. Now, these experiments were done with high levels of commercially available artificial sweeteners, very far from the toxic levels. Nonetheless, these were high. And to move a little bit forward, we wanted to kind of test them in their purified form, in lower levels, in other conditions. And we chose to focus on saccharin because of all the 3, it is the only one that was available in affordable prices in the quantities that we use for these really giant experiments. So from now on, we focused on saccharin as an example. So we utilized purified saccharin, and we repeated this experiment and in many different doses, stemming from the very low doses to intermediate and high doses. In all of them, we saw that it induces a very significant rise in blood sugar levels as compared to vehicle, to water in this case. We then tested the same experiment in different dietary regimens, for example, high-fat diet versus low-fat diet, and we could reproduce the same phenotype in different dietary approach. We tested this in different strains of mice to see that genetics was not affecting us in our results, and in other strains of mice, we again saw that saccharin was inducing the same effect. And in all of these many experiments, when we gave broad-spectrum antibiotics, we could completely cancel out these hyperglycemic saccharin-induced effects. So this now became very interesting to us. So if saccharin does something that is mediated through the microbiome, then we would expect that the microbiome would functionally or compositionally change following the consumption of saccharin. We are very good in characterizing the microbiome in many deep and diverse multi-omic manners, which we did. And the take-home message is that we found in every facet of microbiome composition and function, you can see here an example of composition, of function, of pathway analysis, that the exposure of mice microbiome to saccharin induced a very substantial set of differences that were actually very consistent along the different conditions, different strains, different diets that we were testing them across. And together, our analysis suggested, as you've heard before, that a big impact of this change was influencing what we call energy harvest, which is the influence of the microbiome on extracting energy from food and transferring it into the host, which could explain some of these phenotypes. Now, all of this was really interesting, but it still didn't prove anything about causality. I've not shown you a single experiment that proves causality by which the microbiome is actually doing or inducing the phenotype, the metabolic phenotype in mice, as you may expect. So in order to prove causality, what we do in the microbiome field, we use a system called germ-free mice. These are mice that are raised in these specialized isolators. This is our own large-scale germ-free facility, and these mice are completely sterile. They do not have any microbiome of their own. Okay? They don't have any microbes in their mouth, in their gut, in their urinary tract. They're completely sterile, and we have a whole team of people kind of running around them and making sure that this is the case. Now, these mice are very useful because you can compare colonized to germ-free mice and see what the microbes may be doing. But they are more useful than that in contexts in which you can transfer into these sterile mice microbiome configurations from different disease settings and study them mechanistically for causation, which is exactly what we did in the experiments I'm showing you now, in which we exposed mice to either saccharin or to control, to glucose or to water, and then we took the microbiome from these two groups of mice and we transferred them into germ-free mice and then measured what happened in the recipient mice that were never exposed to saccharin. They never saw any artificial sweetener. And the results were an exact phenotype of the phenotype that I've shown you before. Either with commercial saccharin or pure saccharin, the exposure of recipient mice to a microbiome that was previously exposed to this agent was associated with an increase in the glycemic response of these mice. And this already proves some causality. We wanted to take this even a step further, and in the next set of experiments, we took a naive gut microbiome, we anaerobically cultured it in specialized conditions that we developed at the lab, and we cultured it together with differing doses of saccharin. So in this case, the saccharin was never inside an animal. It was cultured ex vivo in the presence of a gut microbiome, and then we took the gut microbiome at different stages of culturing and transferred it into germ-free mice, and you can see that again, we could induce the same hyperglycemic surge upon the transfer of this saccharin cultured microbiome. So these were kind of experiments done in mice. You know, we like them, they're very interesting, but I think what interests the world in terms of the potential implications of this is what about humans? And of course, studying this in the context of humans is extremely difficult and complex, and what we've done is a small-scale pilot preliminary study in which we tried at least to scrape the surface and address this issue. So what we've done is we've taken a small group of 7 volunteers. It was actually quite difficult to find volunteers that have never been exposed or consumed any form of shape of artificial sweeteners, at least in Israel. I'm sure this is also the case here. And these volunteers agreed to be excessively tested during using all these readouts that I've shown you in the personalized nutrition project, including a daily microbiome assessment. And after 2 days, they started consuming commercial saccharin in a high but not excessive dose. So it's the equivalent of 8 sackets of saccharin a day in Israel. So it's high, but it's not, you know, at least in my view, it's not, you know, super physiological. And we, of course, excessively tested them with a continuous glucose monitor. And what we were surprised to find that we could group these individuals into 2 subgroups. Some individuals didn't care about the consumption of saccharin in terms of their glycemic response. They remained completely stable. While around half, 4 of the 7, developed after a few days of exposure a quite marked level of hyperglycemia. You can see the cumulative averages of the responders versus non-responders in terms of the comparison between their starting and endpoint glycemic responses. And what was really interesting for us is that when we measured the microbiome on a daily basis, you can see that the non-responders maintained this really stable microbiome configuration throughout the follow-up period, while the responders started from a different microbiome and it was much less stable than the non-responders. Now, of course, again, this is a nice observation, but it tells you nothing about causality or causation. And again, we are able to use the germ-free system as a transfer system that would also, at least to some extent, provide hints of causality in the human setting, which is exactly what we did. We took from these volunteers, from each of these volunteers, their microbiome from the beginning of the test and from the end of the test, so before they were consuming saccharin and after a few days of consumption of saccharin, and we transferred each of these microbiome into groups of germ-free mice and measured what happened in the mice. And you can see here an example of 2 of the non-responders. And now we're looking at the mice. We're not looking at the humans. We're looking at the transplanted mice. And you can see that in these non-responders, when we transplant the microbiome from the day 1 or day 7 humans, the glycemic response is quite identical. So there is no change in the microbiome response between these 2 days. However, when we do the same transfer from the responders, you can see that on day 0, we have a lower glycemic response, while at day 7, the transfer induces in the recipient mice a much higher and significantly higher glycemic response, suggesting that at least part of this responding or hyperglycemic phenotype in human may be mediated in one or another ways by the microbiome. Now, this is a pilot, a small-scale pilot, but I think it gives us a nice beginning to study this more extensively, as I will show you in a second. Now, of course, this study created a lot of interest, as you may imagine. What happened in the last 4 years is actually a rain of mouse-based studies, all corroborating and validating our data, linking different artificial sweeteners to the microbiome, to downstream cardiometabolic phenotypes in mice. So in mice, you know, this is all that fit in this slide, but there are many studies that came out by many groups, of course, making us very happy. In humans, as you know and as you've heard in the last 2 fascinating days, you know, the situation is much more messy, and you know, there are studies here and studies there and meta-analysis, and there's a big mess. And if I were to summarize, you know, our 2 cents on this, you know, the jury is still out there. But you know, the 3 major limitations from our angle that we think have not been properly addressed are, one, that none of these studies address the microbiome. Okay, so that's one thing. The second thing is that none of these studies address the personalized aspect. So everybody uses, you know, complete averages and standard deviations, but what our data suggests in the Personalized Nutrition Project, and in this subset of the Personalized Nutrition Project, it's also true for bread, for whatever food we test, is that some people react differently than others. And this has not been addressed. We're not exposed to the raw data of all the numbers in all of these studies. And the third aspect, which is a great limitation, is the readouts themselves. All the readouts related to glycemic control are based on either hemoglobin A1C or GTT, which are good, we like them, but they're extremely noisy and limited parameters. While using the continuous glucose monitoring system gives you, instead of 6 data points for GTT, it gives you 5,000 data points, which gives you a greater resolution in terms of postprandial glycemic response, which is probably the major component here, and many other parameters. So to kind of help contribute to this, we have embarked in our own prospective double-blind placebo-controlled study, which features around 100 individuals, and actually the collection of the data from these individuals has just finished. This created a huge amount of data, so it will take us a little bit more time to analyze, and I promise you that we will publish it, whatever it is, we will publish it. We're not part of this fight with artificial sweeteners, but you know, we feel that this is our duty. But on a higher level, I think that these studies and studies by others kind of point towards the human microbiome variability as being a neglected but very important feature which may explain many of these variabilities in clinical phenotypes which you encounter in these and other questions. And we have computational tools today that enable actually to harness the microbiome in explaining these variabilities and even predicting them. So for example, for artificial sweetener, we easily developed a predictor that enables to take a microbiome of even these 7 individuals and to predict who would be a responder and non-responder even before they consume the microbiome. And I imagine you would be able to do this much better with larger cohorts and deeper dive into the microbiome. So I think it's actually a promise and something to look forward to in the future. And I will finish by thanking the many great people in my lab coming from 4 corners of the earth who are doing these projects. We're always looking for more good ones. I will thank my collaborators, the funding agencies that are making our work possible, and I'll stop here. Thanks.